There are 3 molecules that are more specific than ibrutinib that are all making their way through phase 1 to phase 3 trials that I discussed. The first is acalabrutinib. The next one is called BGB3111, and the third is ONO4059. Acalabrutinib is the most advanced. It's in 2 ongoing phase 3 registration trials. And both acalabrutinib and BGB3111 have very carefully optimized their binding to BTK. They bind to the same site as ibrutinib with a covalent bond, so very similar to ibrutinib in that respect. But they do not inhibit ITK, which may allow them to interact more favorably with monoclonal antibodies, but may also reduce their beneficial effects on the immune system that have been suggested for ibrutinib. And they also have, at least acalabrutinib has less activity against TEC, which means that there's potentially less bleeding. And so what one might expect from these drugs is less toxicity. Efficacy, it's really too early to tell. They're very highly effective. Response rates are almost universal, and there have been very few relapses, but the follow-up's about a year. Then in the PI3 kinase inhibitor area, there are 2 drugs that are in registration trials. They both inhibit PI3 kinase delta. The first, duvelisib, also inhibits PI3 kinase gamma, so it can also hit T cells and neutrophils. And that has had high activity in CLL in the phase 1 setting, about a 60% response rate in patients with a high likelihood of 17p deletion, and the 2-year PFS is about 60%. That's in a registration trial versus ofatumumab that may read out later this year. The other drug, TGR-1202, is an interesting drug. It hits delta, and it's recently been described to have another target also, casein kinase 1 epsilon. It has had to go to very high doses and reformulate to achieve good PK in patients. And so the number of patients on those doses and the duration is still relatively limited. They have seen high response rates. I think durability is still pending because of the short follow-up. The toxicity pattern does seem to be different. It does not seem to have the degree of autoimmune toxicity we see with the other delta inhibitors. And whether this is because it's not hitting the target as effectively or because it has other targets like casein kinase remains to be determined. Now, I also mentioned SYK as a potential target in CLL, also in the BCR pathway. I spoke a little bit about entospletinib, which is a drug in development that targets SYK, which had data presented at ASH in the post-BTK and post-PI3 kinase setting. In that setting, the response rate was only about 25% with a PFS of 6 months, which is not great, but the patients are very sick. And the way in which this drug will be developed remains unclear, although it's an interesting target. And then very interesting data also came out in Richter's transformation that both nivolumab and pembrolizumab seem to have significant activity, at least among a handful of Richter's patients treated on trials, one at Mayo and one at MD Anderson. And so there are larger trials planned with those checkpoint inhibitors in that setting.